Okay, guys, well, welcome back. Sorry for the sudden abrupt ending of the last video. Welcome back to part 3 of looking at cell origins. So we're kind of telling the story, really, aren't we, of where these cells have come from. And we're talking now, looking back way millennia ago, and we're looking at how our first cells originated. So we said there were some very basic gases floating around. That was these, okay, with hydrogen, nitrogen, water vapor, methane, ammonia, hydrogen, sulfide, all these coming out of volcanoes. Okay, but at the time they were just forming single molecules, so these molecules weren't joining up in any specific shape or form, they were just floating around willy-nilly in the environment. Now what they think happened at the next point in time was that the monomers, or what we're looking at, these are what we call monomers, just single molecules. Okay, what happened is they started to mix within what we call the primeval soup, which are the shallow oceans which were around at the time. And they begin to form into more complex biological molecules. Now, there was a very important study done by a couple of scientists called Miller and Urey, where they tried to recreate the conditions that made this happen. And this happened back in 1953. And they were trying to prove this idea of chemical evolution, whereby really complex molecules could form from very simple ones in this kind of idea of a primeval soup. And so they combined the molecules from the previous page that we looked at, the hydrogen sulfide, nitrogen, water vapor, all those different things in a closed glass vessel, which constituted their kind of simulated atmosphere, or what they thought the Earth would have been like in its atmosphere in that time. They heated the water up that was inside it to simulate the volcanic activity, and they sparked electricity through the gases to simulate lightning. Okay, and they found some pretty incredible things. Now, before we actually talk about what conclusions they made, we're actually going to look a little bit more closely at the experiment itself and how they did it and what they did. So bear with me a minute. So this is a basic kind of model of what they designed, how they kind of made their setup, and it's pretty awesome. So what did they do and how did they do it? So obviously here you can see a heat source. It's got water running through it. They've got an inlet valve for certain things. They've got a gas inlet valve. They've got a cooling system. They've got electrodes to spark it. They've got a heat source to heat up the water. Okay, and these are the main things that they've got in it. So the conditions that they put it under included a reducing atmosphere, so it was very low oxygen, high radiation levels, high temperatures, and electrical storms. So the temperatures came from here, and the electrical storms came from here. The water was boiled to form a vapor, and then was mixed with methane, ammonia, and hydrogen, which was inletted here. The mixture of gases then went obviously through here and was exposed to the electrical discharge, or sparks, to simulate lightning. And then the mixture was then allowed to cool, and after 1 week later, they sort of analysed what was coming out of it, analysed the chemicals that was coming out of it, and they found some really interesting things. All right, they found that some very simple amino acids had been created. Okay, so they'd actually made some new chemicals, or them basic amino acids, and as we all know, amino acids are necessary for polypeptide formation of proteins. Okay, so that's a really key idea. They also found that some very complex oily hydrocarbons were formed. Okay, so things that were quite flammable. All right, which is also quite interesting. Now, of course, based on these findings, it was concluded that under the hypothesized conditions, so these are the sort of hypothesized conditions of what they thought the Earth was like, organic molecules could be formed. So these carbon-containing molecules, carbon-rich molecules, like amino acids, could actually be formed under the right conditions. So after that week, they found that 13 of the 20 naturally occurring amino acids had been created. Now remember, there's only 20 naturally occurring amino acids, and 13 were made in this experiment. So that's pretty awesome. Now, unfortunately, Miller and Urey's experiments allowed for the formation of amino acids, but the conditions used also tended to hydrolyze bonds, preventing any polymers forming. So they were making these lovely, interesting amino acids, which of course are massively necessary for life, but there was still no explanation for how these amino acids were getting joined together to form polypeptides. And so it was a while later, or quite a long time later, until they figured out that there was a mechanism for the way these polypeptides, or for how these polymers or repeating units of chemicals were actually being formed. And the answer lay in deep sea thermal vents, which is what you can see here. Okay, so this is the vent underneath the water. They lie in pitch darkness at the bottom of the ocean. No sunlight can penetrate these areas. Okay, and they spew out these vents of sulfur-rich gas, okay, which you can see being kind of ejected into the water here. And you wouldn't believe it unless you saw it, but these actual areas are teeming with life. There are crabs and sponges and things which live around here, all right? And they're surviving, and they've survived for millions of years in the absence of light. Now, before this was discovered, people thought that light was the energy source for the original energy source, or sunlight was the original energy source for all life on Earth, okay? And this completely threw that theory out of the window, okay? Now, obviously, sunlight is very, very important for life above the oceans, and for most of the life in the oceans, but we now know that life can survive without it. So why else were these deep sea vents important? What else were they doing? Why else are they so cool? Okay, so the fissures in the planet's surface from which this water is coming out of are traditionally found in volcanically active areas. And now, along with heat energy, the vents issue a ready supply of reduced, okay, so very oxygen poor, inorganic chemicals, so chemicals that don't have much carbon in them. Now, what these vents do provide are the right conditions for chemicals to allow organic polymers to arise. So with that heat and with the right mixture of chemicals, okay, it allows chemicals, different polymers, or different monomers to join together and to bond together to form these long repeating chains. And that is pretty amazing. So again, along with, so the primordial soup was forming our organic compounds, and then our organic compounds were being joined together in these areas, okay? So that what would have to happen is these organic compounds formed in the shallow primeval soups were going to have to sink down into the deep oceans around these fissures, and that's where they'd be joined together to form these long repeating chains of molecules, okay? So that's where we think something happened there. All right, so very interesting indeed. I'm sure you think so too. Okay, so we've got the formation of our organic molecules like amino acids. We've got the formation of our polypeptides being done at the bottom of the oceans in these deep sea vaults, but that still doesn't allow us to recreate or enable inheritance or self-replication of these molecules. So how did that happen, and whereabouts did that come about? All right, so DNA, which is what we looked at before, is a very, very stable and is very effective at storing information and is not able to self-replicate, okay? Enzymes are needed to self-replicate DNA, okay? That's something that's very important to remember. So DNA alone isn't able to self-replicate. However, RNA, which is made from DNA, can both store information and it can self-replicate itself. So it can catalyze the formation of copies of itself, and that is immensely important. In ribosomes, which are these little small organelles which you find in nearly all cells, in ribosomes, RNA is found in the catalytic site and plays a role in peptide bond formation. Okay, so they think now, and we're talking about this, they're thinking now that the RNA, when it was made, was the point in evolution whereby it was able to start self-replicating its own stuff, and from RNA build new DNA. All right, and that was the point at where it all kind of blew up and became more important, and all the different types of organisms kind of evolved from that point. Okay, and that's really, really important to remember. So the next part we're looking at is the formation of membranes to package organic molecules. Now, obviously, we've already looked at the structure of membranes. We know that they have a phosphophilipic head, so they're actually attracted to water, and a hydrophobic tail, which are these bits, non-polar areas in the middle of a cell. And so because water, remember, is polar, the water is attracted to the heads, okay, but is repulsed by the inside of the cell. So what essentially happens is it forms a barrier between the water and whatever else is inside the cell. All right. So at the moment, scientists have been looking at this, and they've actually shown that the phospholipids form naturally assemble into bilayers, and if conditions are correct. Now, the formation of bilayer creates an isolated internal environment. So this is the isolated internal environment. So the formation of an internal environment means that optimal conditions for replication or catalysis can be maintained. So what that means is, so before we've got our amino acids, we've got our polypeptides, and we've got our self-replicating RNA. When these molecules were formed using fat molecules, okay, it provided this perfect area for these reactions or for the self-replication of different organelles. Okay, now this is where our first prokaryote began to emerge. Now, those four-step processes that we talked about occurred over millions of years. It wasn't just something that happened in a day or two. Okay, it happened over millions of years. Okay, guys, we are nearing now the end of our video lecture on this topic, so just bear with us for a second. Now we can look at, so we've got the formation of the membranes around the different chemicals. Now we've got to look at the formation of the nucleus and where that developed from and how it developed. So here's your ancestral prokaryote. Now a prokaryote grows in size and began to develop folds in its membrane. So you can see these folds forming here, these little cracks in the membrane as it starts to push through. Okay, kind of like what happens in endocytosis. And it starts to do this because it's trying to maintain a really, really efficient surface area to volume ratio. After a period of time, the infoldings become pinched off, forming this internal membrane. All right, so this is not pinched yet, but over here you can see it begin to become pinched inwards. Okay, the nucleoid region is enclosed in the internal membrane and becomes the nucleus. Okay, so this is the nucleoid region that we can see inside here. Okay. All right, so that's the nucleus. Now we've got to look at how the origin of the eukaryotic cell can be explained by the endosymbiotic theory. So over here was the formation of our first prokaryote, as it were. What happened next? Okay, so here's your ancestral prokaryote. You've got the infolding of the membrane. It's already pinched off to form your nuclear membrane and your nucleoid here. So what happened next? Well, first of all, we get an aerobic proteobacterium. So a fairly advanced anaerobic bacterium comes along. Okay, it could have been entered into a larger anaerobic prokaryote. So here is your prokaryote here. Here is your aerobic bacterium, okay, entering through the membrane, possibly as a prey. It might be eating it, okay? And the same possibly with a chloroplast up here, with a cyanobacterium, which were the initial photosynthetic bacterium that came about. Now, if it survived digestion, it could become this valuable endosymbiont, which basically means this is going to start producing energy for this. Okay, and in the same way, this cyanobacterium begins to absorb sunlight, okay, and make sugars, valuable sugars that this bigger bacterium can use. So the aerobic proteobacterium provides a rich source of ATP to its host, enabling it to outcompete other anaerobic prokaryotes. And as the host cell grows and divides, so does the aerobic proteobacterium. Therefore, subsequent generations automatically contain aerobic proteobacterium. So we're getting more and more generations of these appearing. The aerobic proteobacterium evolves and is assimilated and becomes eventually, through millions of years of evolution, it evolves into our mitochondrion, and this one evolves into our chloroplast that we find in plant cells, which is pretty awesome, eh? Okay. coming very close to the end now, guys. I know, thank you for bearing with us. It's been a very, very long video. So the last one is looking at the origin of eukaryotic cells and how it can be explained by this endosymbiotic theory. The evidence supporting it is that, first of all, they have the evidence supporting the endosymbiotic theory for mitochondria and chloroplasts is that they have their own DNA, which is naked and it's circular. But when I say naked, I mean it isn't associated with proteins like our own DNA is. Both mitochondria and chloroplasts both have ribosomes, but these ribosomes are different to ours. Again, they're smaller, or what we call 70S ribosomes, which are much smaller than the 80S ribosomes that we found in our cells. Okay, outside of the mitochondria and the chloroplasts. So again, that's more evidence suggesting they came from bacteria, which all have 70S ribosomes. They have this double membrane, so you can see how the membrane in these things is double, okay? And an inner membrane has proteins similar to that of prokaryotes or similar to that of bacteria. Again, they're much smaller than our normal animal cells, being similar in size to bacteria, okay? And they're very, very susceptible to the antibiotic chlorophenicol, okay, which is an antibiotic which many bacteria are very adversely reacted by. They transcribe their DNA and use the messenger RNA to synthesize some of their own proteins. So they're not reliant on our nucleus or the nuclear DNA to make proteins for themselves. They can do that by themselves. And they can only be produced by division of pre-existing mitochondria and pre-existing chloroplasts. So that's the only way these can be reproduced. They're not synthesized from proteins like the rest of the organelles, like the Golgi apparatus, the endoplasmic reticulum. These are all synthesized from proteins that we make from DNA and RNA. Okay, that's not the case with mitochondria and chloroplasts. Now, 17 minutes long, guys. It's time to stop the video. It's a long one. Thank you so much for listening, and remember, keep making those questions if you don't understand things. I know there's a lot here today. Thank you very much. Well done.